diploid quality of chromosomes. Diploid meaning when there is double the amount of chromosomes in the cell, as you can see here. Meiotic division splits the cells in order to begin the primary spermatocyte. We still have a diploid number of chromosomes, which in humans consists of 23 pairs or 46 chromosomes.
Meiosis occurs at the primary spermatocyte. This involves cross-linking to enhance variation. This is further echoed by the inclusion of random alignment, which sees the chromosome's orientation and positioning being randomly differed each time.
After the cell division, gametogenesis continues, with spermatids being formed, continuing a haploid number of chromosomes.
As you can see, this unique cell division is concluded with spermiogenesis, forming the spermatozoa, also referred to as the male sex cell. Of course, for an AS level qualification, only the simplified view of spermatogenesis is studied. However, it can get extremely complex and complicated when studied at an advanced level.
This is the male gamete, also known as the spermatozoa. These are usually 50 micrometers long. In order to be transported through the female reproductive tract, they must remain in semen.
The structure of the sperm starts at D and E, with E being the acrosome, which contains digestive enzymes to break down the surrounding layers of the ovum and the sperm's head, so that the sperm is able to penetrate the egg. At D, we have the nucleus, which is also in the head and contains a highly condensed haploid number of chromosomes. At C, we have the middle piece of the sperm, which contains mitochondria used for energy.
The tail is called the flagellum, which propels the sperm by its movement in a liquid environment.
Today we are going to be describing the sections of the ovum. The human ovum is about 0.1 millimeters across, which in comparison to other animal eggs such as the ostrich is considered small. The corona radiata or follicle cells form part of the protective barrier of the ovum. Inside of that, you'll find the zona pellicuda, which is a protective layer of the ovum that initiates the chromosome reaction. The cytoplasm of the ovum contains many food reserves. The nucleus contains the nucleolus, and the nucleolus contains the genetic information needed for fertilisation.
Oogenesis is the formation of ova.
Each primordial germ cell in an ovary results in only one egg.
The number of eggs produced in a lifetime is lower than the number of sperm.
The primordial germ cell divides by mitosis to produce oogonia, which then divides again by mitosis to produce a primary oocyte. As this divides by mitosis, the primary oocyte has a diploid number of chromosomes.
The primary oocyte then divides by meiosis to produce a secondary oocyte with a haploid number of chromosomes. This is the only egg that is released by the ovary and can only be completed once fertilised. A polar body will also be produced.
A second meiosis division occurs, producing 3 polar bodies that will die, and only one will survive to become an ovum. Variation in meiosis occurs through random alignment and crossing over.
All the genetic material is invested in one ovum, therefore it would not make sense biologically to waste resources by producing too many of them.
Acrosomal reaction occurs when the spermatozoa reaches the ovum.
As we've previously learned, the sperm has a membrane-bound site of enzymes called the acrosomal enzymes, which are contained in the head of the sperm.
These enzymes are released as the sperm touches the ovum.
The ovum that is released during ovulation is surrounded by a protective jelly-like layer called the zona pellucida, which is also surrounded by follicle cells.
These enzymes digest the follicle cells and zona pellucida. However, one sperm does not contain enough acrosomal enzymes to get all the way through the protective layers, so it requires more sperm to break down the layer.
Now the protective layers have been broken down, the sperm can now reach the ovum.
A sperm cell reaches the egg. This triggers an acrosomal reaction.
The head of the sperm then begins to push through the gap in the zona pellucida.
The head of the sperm reaches the cytoplasm of the egg cell and meiosis is complete.
The head of the sperm cell then enters the cytoplasm and the cortical granules harden.
This stops any other sperm from entering the egg cell. This would cause an extreme mutation. This is called polyspermy.
The chromosomes of the sperm and the egg are now free to undergo mitosis.